Ladies and gentlemen, welcome to the second day of the 2020 webinar series on crystallization, organized by the Working Party on Crystallization of the European Federation of Chemical Engineering. Working Party on Crystallization. And I have the honor and the pleasure to host these webinars on behalf of all my colleagues. The European Federation of Chemical Engineering, the EFC, is a non-profit making association whose objective is to promote cooperation in Europe between professional, scientific and technical societies in 30 countries for the general advancement and the development of chemical engineering. The Working Party on Crystallization consists of crystallization scientists and practitioners from academia and industry. It advances the theory and practice of crystallization. It organizes the triennial International Symposium on Industrial Crystallization called ISIC, as well as other workshops, including training events. And it promotes exchange about standards, protocols, and best practices within the crystallization community. You can access the Working Party on Crystallization through different platforms and social media channels that are reported on this slide. I'm also obliged to make clear that the views expressed by speakers or third parties during EFC's webinars are those of the speaker and the third party, and not necessarily of the European Federation of Chemical Engineering or of the institutions associated with those individuals. Today's 2-hour webinar is devoted to the fundamentals of crystallization. It will be chaired by Professor Heike Lorenz, who is a professor at the Otto von Guericke University in Magdeburg, as well as scientist at the Max Planck Institute for Dynamics of Complex Technical Systems in Magdeburg. She will be the co-chair of the next ISIC conference to be held in Potsdam in September 2021. So Heike, the floor is yours.
Yeah, thank you, Marco. Good afternoon also from my side, and welcome to the webinar series of the Working Party, EFC Working Party on Crystallization. It's me a pleasure to chair this second session, second day of this webinar, which is targeted on fundamentals of crystallization. We will again have 4 lectures, 1 keynote lecture and 3 shorter ones, all focusing on experimentally based crystallization studies connected to modeling and simulation, aimed at a deeper understanding of crystallization processes. The talks will touch crystal shape determination, polymorphy and polymorphic transitions. We will see something about the new novel in situ X-ray microscopy technique, and also a new method for scale up of precipitation processes. The keynote lecture will be given today by Heiko Briesen, and it's me an honor to briefly introduce him. He is Professor of Process Systems Engineering at the School of Life Sciences of Technical University Munich in Germany. He received his PhD from the RWTH Aachen, and his research interests are very broad. In general, modeling and simulation of complex processes. So crystallization is only one of those, and the objectives of his group's work is to develop and to transfer process systems engineering concepts to food and beverage technology and biological processes as well. So meaning using mathematical and computer science techniques to both develop new and optimize existing products and processes in the related industries. So Heiko, we are looking forward to your talk. The floor is yours.
I cannot start my video. Ashwin, you should let me start my video. Hello. Now we go. Here we go. Hello, everybody. Let me share my screen. This should be the one. And start the presentation. So the last thing I should do is switch on the laser pointer I was told. So here we go. Hello, everybody. It's a great pleasure to give this presentation here. Thank you, Heike, for this kind introduction. I will mainly talk about crystal shape today, as it is here on the title. First question would be, why should we bother with crystal shape at all? I mean, when you look at these nice sodium chloride particles, crystals, they're perfectly cubic, and crystal geometry could be so simple if each and every crystal would look like this. This is unfortunately not the case. We have to deal with much more complex systems. The harsh reality looks like this. For example, for a paracetamol crystal, you see here, or a lactose crystal, this classical tomahawk form, or here another, and the last one, a potash alum crystal here. Note that in this, for example, in this potash alum crystal here, all the phases which could potentially arise are given here. This is not the typical complexity we are facing when we have potash alum crystals. But typically, they look more like this perfect octahedron here. However, there we have other problems here of non-ideality, for example. You see here that potash alum crystals can show such sort of non-symmetry here, which you would not expect, because all the phases here should usually grow with the same speed, with the same velocity. So you should, in theory, not get this kind of non-symmetrical particles, however you observe them. Also, you have to deal with agglomerates, which obviously also look very much different from these perfect cubes or perfect spheres. Still, I have to click here, still in modeling or in representing it for experimental values, you often only use a volume value and consider a volume equivalent sphere or volume equivalent cube as a more or less correct representation of your crystal. And I think when you look at the comparison of the crystal variety you have and on this type of representation, it almost already becomes clear this is probably not the best way to go. There are kind of loopholes. You try to introduce aspect ratio or sphericity values. However, there you run into other problems because sphericity and volume is not unique. You can have very, very different particles or crystals with the same sphericity or same volume, elongated. I don't know if you see them here. I try to put them in the camera. These are two crystals. I use crystals, these are aluminum pieces, which I use in class to show this problem, this fundamental problem of shape characterization, because both these shapes you see here, this needle one and this plate one, have the same volume and have the same sphericity. And you basically run into this kind of problem with any sort of lower dimensionalized characterization. This is hard to avoid. So on the side of the product and on the processing, crystal shape has also several effects. For example, it affects product value or product quality. For example, dissolution kinetics of pharmaceutical compounds are determined by the shape. Also processability, for example, the shape of the crystals determines its filterability. If this slide does not convince you yet, I will show you this slide here. This is a collection of yesterday's presentations. All of these presentations show some crystal shape behavior. Here in Martha's talk, you see this potassium dihydrogen phosphate particles, which have this prismatic form. You see here prismatic crystals. These here, the width have been reported of this very small crystals. These are not the ones I will be talking about. And here in this presentation, we have seen quite nasty agglomerate systems here. So you see that this is basically a problem which you face in almost every kind of crystallization problem. So I realized that when with increasing age, keynote lectures tend to fall into some kind of history mode, and I cannot save you from that. I will go with the flow and give you a little bit of an idea of my personal journey in this crystallization shape. And it basically started out at this conference here. This was a ESIC conference, the 15th one in Sorrento in Italy in 2002. By the time I was was near the end of my PhD and had not worked at crystallization at all. Also, our group had no base expertise in crystallization, but I was looking for other areas to explore. And my professor, Professor Markwood, by the time, was kind enough to send me to a conference where I had nothing to present, where I had no idea about what would be there. And I can tell you with this level of personal ignorance, each and every presentation was totally exciting to me and opened new perspectives on basically each and every presentation. There was one particular presentation which probably resonated most with me, and this was, click here. This was here from the program. This was a session on molecular aspects of crystal growth. It was a plenary lecture of Mike Doherty, where he talked about a model for predicting the shape of amino acids crystallized from solution. So this was two aspects which have been addressed here. One was the molecular world, and one was shape of crystals. And actually, we picked up both these areas in our research. We do some extensive work in molecular dynamics modeling, which I will not talk about today. I will only focus today on this shape aspect. Another thing to realize is probably that when you look at the date here, this is almost exactly 18 years ago. And in Germany, 18 is basically the legal age for everything. So I consider this to be kind of to give this presentation here as a kind of a birthday present to me from the organizers, which I want to thank very much again for giving me the opportunity to wrap up our research here of the last couple of almost 18 years. Some small disclaimer should be in order. I will very much focus on our own work. There is much excellent work out there, but I decided instead of mentioning only a few of them and make the other ones mad at me, I thought I'd better mention none of them and equilibrate the level of madness of the people, and hope you forgive me if I do not refer too much to other work, but focus on our own. Let me first try to decompose the problem we are looking at. There are different approaches you can take when you look at crystal shape. You can have a complete modeling and simulation view, or you can look from a very experimental perspective. In terms of geometry, you can basically climb up the ladders of dimensions here from a one-dimensional characterization, which basically would be only characterizing a crystal by its volume, and you can climb up the ladders here to a full three-dimensional characterization. There is sometimes misunderstanding between three-dimensional characterization and three-variate representation in terms of population balance, for example. Of course, also a perfect cube is a three-dimensional object and a three-dimensional representation, which, however, can be totally described by 1 degree of freedom, a side length. And also, on the other hand, there are complex geometries which you need more than 3 degrees of freedom to characterize a three-dimensional object. So this probably I will not be perfectly consistent throughout the presentation in this issue, but I hope this becomes clear from context. Another thing to worry about or to consider is, do you look at a single point in time, for example, at the end of an experiment, or do you want to track a time evolution? Usually for modeling, for example, you usually run with a time evolution by its own because you have the model. In experiments, this can be a large effort to track a time evolution. And the last issue to consider here is what I would call connectivity somehow. This is, do you have the option to study your process online and inline, or can you study the process only offline? So this is basically the menu from which we should choose from. And the question now is, what do we like to order? So in the optimal case, I would say this would be our perfect world. We would like to have a consistent framework for modeling, simulation, and experimental characterization up to the full three-dimensional characterization for a full-time evolution set, and we want to have this online. This would be our wish list, so to say. So as the go with the Rolling Stones, you can't always get what you want, and forgive me right now, this will be not the last rock music reference in this talk. So if you get bored by listening to the crystals, you may start counting the rock music references. There will be a solution at the end of the presentation. So this will be our wish list here, but this is kind of wishful thinking, which is, by the way, another rock band from the 70s. And what we like to do, what I want to show you in this presentation is how we can climb up this stairway to heaven, so to say, and in the end, get most closely to this state that we have most of these readily framed issues here resolved. So let's try to take the first steps on this stairway to heaven, and I will go with the first work we did, or I did in this context. My first work was with regards to modeling. So going back to these icons here, you will see these icons throughout the presentation a couple of times, so you can keep track on what kind of concept I'm talking about at the particular moment. So our first steps have been in modeling, and obviously then time trajectory is not an issue. You model the time evolution, and in our first steps, we went from a one-dimensional population balance characterization to a multivariate characterization, which should be able to capture shape in some sort of way. So first thing would be similar as the two aluminum pieces I just showed you, was not to consider cubes, but needles or platelets, but in this case, where at least two of the side lengths were identical. So a two-dimensional characterization, but you still have the complete options of getting plates and needles by phase-specific growth rates. So, and this was a kind of result here. The basic contribution was here not the standard forward simulation, but a kind of a model reduction scheme to go from a truly two-variate population balance to a set of three monovariate population balance to reduce the computational effort. I would say by the time today, two-dimensional population balance models would not be considered to be too computationally demanding. By the time this concept of model reduction was still hopefully quite interesting. Another work here, which was shape related, also population balance model, was the interplay between crystals, of growth of crystals and attrition of crystals. So when you consider this type of perfectly cubic crystal here, if it gets collisions with a stirrer, you chip off some edges probably and arrive at some more rounded version. This is the most rounded version you can get in this type of representation here. And for this, there is a quite interesting, quite interesting interplay. I don't know why I go back one more time because I don't know why the animation is not running. I hope it runs now. Here we go. You see here, this is now the bivariate evolution of a particle distribution function. So what you see here is the increase of attrited secondary nuclei which grow, and this, you see here that this axis here scales, so you arrive over time at some similar solution, which does not change anymore, which was quite interesting, I think, at the time. And there is a nice interplay between shape and the process, because it's not only that the process, the attrition, does change the shape, but also the changed shape has an effect on the attrition. You can easily expect that a very sharp-edged crystal like this one here is stronger affected by a collision with a stirrer than a more rounded one. And this interplay here leads in the end to this kind of self-similar solution. So in the next step, I want to go up the dimensions here, and I look then at aggregates or agglomerates to see something like this here. You can really, in a full three-dimensional way, glue together primary particles to construct agglomerates here. You can do this also with plates and needles. And with that, you can arrive at something like that, which looks like artificial micrographs, which you could, for example, compare to classical microscopic two-dimensional images here. From that, when you look very closely at these pictures here, you would see that there are gaps between the particles. This is because by the time I did not have the concepts ready that a particle overlap could actually be addressed properly. So this has been done that in the later years here by students of mine, Jana Kovacevic and Alexander Reinhold, to develop most of this stuff here, that we can actually decompose this sort of aggregate into the primary particles and subtract here these kind of volumes, which are the overlapping volumes, to get really the detailed kind of volume of the crystal. With this kind of concepts, we did then rerun of several simulations here. And one thing we did, we tracked, for example, the total volume, which was governed by growth. And when you consider this type of crystal here growing, you could easily expect that, for example, these faces here, which are glued together, will no longer grow. So they will not deplete any supersaturation. They will no longer contribute to the volume change. So this is reflected in this curve here. If you do not consider this, if you would go with a single shape factor, as said, this is the loophole which you can go for, you would end up with this here. This would be the best shape factor you can get for this type of agglomeration. You see you're way off. Same is true for the total area, which is basically the reason for this, that you're way off with the volume. And here again, we developed then a two-dimensional population balance model and could nicely show that at least the two-dimensional model for this type of complexity, for this potash alum octahedral particles here, you can nicely represent this only by volume and surface area. But the bottom line is, if you have aggregation in your process, one-dimensional population balance, if you're not dealing with coalescence of liquid particles, liquid droplets, then a one-dimensional representation will probably not do the job. This is the bottom line of this slide here. So now that I've did some work in modeling, I discussed this a couple of times also with colleagues. I guess some of you may know Dirk Wieghuysen from Novartis, whom I tried to convince a couple of times on the beauty of modeling and simulation. And he's a very passionate experimentalist, and he pushed me then into the idea of really looking at crystals. So what options do we have in crystallization for characterization? Well, some standard methods would be laser diffraction. However, for example, in laser diffraction, you cannot get any shape information. You need to provide shape information to get more or less correct size information. So the same is true with FBRM. We did some FBRM modeling ourselves, and you see that when you want to really have to really in detailed way want to construct the signal modeling, you have to provide this shape of the crystal instead of being able to identify it. So what Dirk Wieghuysen always said, look at the crystals, they tell you so much. So the idea was, okay, looking at crystals means imaging techniques. So we dived into this. Now you can do very different sorts of imaging here. Standard would be to have really two-dimensional microscopy, and you can do this offline, and you can also do this online. You see here we have this online button here with a quick pick system, for example, with a bypass, and loop the certain amount of crystals here and get the information also on crystal shape, at least in a projected manner, in a one directional projection. Now then, there has been many excellent work and also follow-up work on this from Marco Mazzotti's group, where they say, okay, we do not look only from one side at a crystal, but we look from two sides, the stereoscopic projection. And from that, you can reconstruct some more information. I will call this here as a 2-point X geometric representation. Still, you're able to do this online and inline to some extent here, which is a very interesting feature. However, we wanted to go for the really full 3-dimensional evaluation, because similar problems as you're facing in the non-uniqueness for this shape factors, which I mentioned before, also hit you in this projections from 1 or even 2 projections. Basically, X-ray computed tomography is doing very, very many projections. What you lose there, and I will mention this later on one more time very explicitly, is the online capabilities. So how will this work? Some of you may know computed tomography from medical devices. There you have an X-ray source and a detector, and both cycle around the patient in such kind of equipment we are using. We do it the other way around. We spin the sample. So this sample here is positioned between the X-ray source and the detector, and you get such kind of a radiograph. And now if you do rotation of the system, if you would do this with a human in a medical device, probably the person would become dizzy, so it's not a good idea. But here, our crystals are very forgiving in terms of dizziness, so you can get many, many projections here. And from these projections, you can actually reconstruct 3-dimensional images. So the term microcomputed tomography, the micro does not refer to the size of the equipment. Unfortunately, this is not a tabletop device. You see here again the X-ray source, you see here the sample holder, the detector, and by varying the geometry, by moving the sample closer or further away from the source and moving the detector, you can generate different resolutions of the system. And you see this is basically, here is a person next to it. This is basically the size of this type of equipment. So what do you get when you try to make such a kind of computed tomography with a crystal? This is basically the base gray value image you get from there. So, but gray value images does not yet, well, when you look at it, you have an idea of what the shape is, but we want to do this algorithmically, obviously. So you need image analysis techniques, and in this case, it's become a bit more complicated because you need 3D imaging techniques. So one of the basic algorithms which is used here is a so-called half transformation, which is used to identify faces in 3D. In 2D, it detects lines, so this is a direct extension. You see here 2 different angles, and these angles here are arbitrary planes in this 3-dimensional image here. And by these peaks here, you basically count how many surface points do you see at a certain combination of angles in a certain plane. So what these peaks here tell you is, for example, you have a plane in this system here, which corresponds to this angle in this direction and to this angle in this direction. So you tilt, flip, and you end up with a couple of faces here. So here, for example, these 8 main faces of this octahedron here are identified. So you see here that there's some stuff which is not completely closed yet. So there's some additional work to be done here. So this is the first step to get this to this basic representation. From that, you use the known Miller indices from your crystal, and you try to rotate such a wireframe model in a way that it corresponds to the actual crystal as good as possible by minimizing its volume differences. And then you can reconstruct also the smaller faces by additionally exploiting the symmetry of the system. So what you end up with is then actually this fully three-dimensional representation of this single crystal here. So in the end here, you get a representation and an identification of all the 26 facets of this potash alum crystal. I give you just some qualitative examples for this. You can end up with this kind of very well-behaved crystals. We have done this with potash alum. We have also done this with paracetamol, where you see here also when you have a very nice crystal here, you get a very nice identification of the wireframe model in really 3D characterization. Well, nicely enough, this system is pretty much robust against small imperfections. So if you have some material grown here at the crystal surface or some imperfections here, still the main shape of the crystal is nearly perfectly represented. Gets a bit more difficult if you have breakage. So when you look at this crystal or this crystal here, you see that here a piece is chipped off and here some material is chipped off. Then the algorithm is able to reconstruct the prior form before breakage of the crystal. So you can even learn something about this this breakage behavior probably. So it is also to some extent then robust against this breakage behavior. A completely different story is when you go from single crystals to agglomerates. So when you have this very simple two primary particle agglomerate and do the same procedure here, you end up with this crystal, which is obviously a failure. This does not properly represent the shape of the agglomerate you see above. So we try to solve this problem again by using several techniques from image processing and push them to a three-dimensional world, and did, for example, here first some, basically in image analysis, this is kind of a segmentation problem. You have to segment an object into several separate objects which are of relevance. This is what we need to do. One classical algorithm for segmentation is a watershed algorithm. So we used here a seeded watershed algorithm, which detects, you see here the color codes here. This would be regions which would be detected. But you also see that this is not perfect here. So you have here the green and the orange part here, which obviously belongs to the same crystal here, have been over-segmented into two different primary crystals. So another thing which is done here, we find this concavity points. Another thing which is important in the tomography, you can not only look at concave, but also on convex crystals and get the complete complexity of the convexity part. So you identify these convexity points where you have an incline basically of two faces here. And starting from that, you can concatenate the different regions by avoiding concatenating over these concavity points. And you arise at this segmentation, which already pretty nicely reproduces this four primary particle agglomerate here in these blue, red, green, and yellow primary particles. For these segmented crystals, you can go back to the algorithm I showed you before, use a single crystal algorithm to identify the primary particles and end up again with a full characterization of your crystal. So then we ask, what can we do with it? We looked at, for example, we had in our samples different agglomerates. These are only agglomerates with two primary particles, and they're obviously different. They have had some different history, how they have been grown and so on. And our question was, how can we identify this, or can we characterize these different types of crystals? So what we did, we identified our two different primary particles, we separated them, took them apart, and then searched away, how do we have to rotate one of the crystals to match most closely to get a congruence with the other crystal. So you have to rotate on two different axes, and the minimal angle you need, the smallest angle of rotation you need, is called what we call the disorientation angle. So when you do this for these two crystals here, two different agglomerates, you would end up here with a quite large disorientation angle. So you have to rotate this crystal here much more to fit or to overlap with this back crystal here than you have to do it for this crystal, where the disorientation angle is very small. So then we looked at a whole set of different crystals and wanted to look how this angle distribution looks like. And there is actually some theory behind it, which has been derived in the midst of the last century already. The red curve, which you see here, is what should be the expected probability distribution of disorientation angles if you completely randomly aggregate your crystals. Then you would end up with this. As we had this Monte Carlo type aggregation algorithms, we run this, and you see that these are the orange samples here. You see that we quite nicely could reproduce this, this theoretical prediction. Now, how does it look like in experiment? We looked at a set of crystals and evaluated this, and this is what we came up with. So there are limited number, so only 45 pairs of crystals, but we hardly observed any larger disorientation angles, and the huge set of crystal aggregates looked similar to this one, so very much oriented towards each other. Short disclaimer at this point, I do not say anyhow that we're able to clarify whether this is caused by preferential aggregation or this is some twinning of crystals. Could be any of those. We just had this observation here at the moment that the disorientation angles are not distributed in a way as it would be expected when you have a random orientation at aggregation. This is all we can detect here. So let me go back to some of the icons. So what we have basically a framework for right now is we have modeling framework for quite detailed characterization of crystal shapes. We can go up to fully three-dimensional characterization in modeling and in experiment. And the next step here would be, can we go from a single point in time to time evolution? The results I showed you before were taken for aggregates at the end of the process. So there's no time trajectory considered whatsoever. We have run the process, took the samples, investigated the crystals. So can we go for more time evolution? So this was our sample preparation in the initial phases of the project. We just throw the crystals into some vials and did this characterization here. And I'd love to say that we have been more sophisticated than this, but this was actually really just a simple toothpick where we have an adhesive tape here, and we basically crumbled the crystals on this adhesive tape and then wrapped the adhesive tape around the toothpick. And this is then a scan you see from that. Why did we do this? One problem here, so basically we have two problems to deal with. We wanted to have more crystals, and we want to have a simple separation of the crystals. So how do you decide if a particle is an aggregate or if it's just touching each other, or it's just separate particles just touching each other when you have thrown them into a vial? So this was a bit of a challenge at the beginning of the project. So we tried to come up with other solutions here, and what I will show you is still sampling based, but we can basically solve both, going to a much higher number of crystals, and also the segmentation, the separation of the crystal becomes much more easy. What did we do? We basically used additive manufacturing and made such printed sample holders here with cavities. And it's pretty painful because it's still pretty painful because at the moment you still have to sort all the crystals with a tweezer into all of these different cavities. But in the end, you can at least stack these matrices over each other. You have no longer a separation problem here. And you can, with a single CT scan, have information on up to 800 crystals. So how does this look like? This is, for example, a CT scan now of a large data set And you see here again our go-to crystals, the potash alum crystals here, which are nicely rotating, which can be easily separated from each other here. So there's no problem with that. We have good contrast of the particles and the surrounding medium because they're in air, and can also have quite a large number, you see here, with one single CT scan. Before this question comes, one CT scan is typically in the order of several hours. It depends on the resolution you're looking for. So again, now we are at a status where we can look at many crystals, but this when you look carefully into these rotating crystals here, we choose a process here which did not produce much aggregation. So we're looking only at the crystal growth at this part. And so what we were interested in is here is how does shape evolve over time, and to do a categorization of the different shapes here, we choose this representation here. So you have from the center of the crystal, you have a certain distance to this face. And you can do this for any of the faces. For a perfectly octahedron crystal, all this H1 to H4 should be absolutely identical. So whatever you do, this should be the same value for any size of the crystal. What we did to get rid of the size information and to focus only on the shape information, we divided each of these numbers here by the largest H value of this set. So you always get a 1 and then a couple of values smaller than 1, probably as close you are to the octahedron shape, they should be all similar to 1. So, and what we observed then, that there are different shapes in our sample actually. So we considered ideal, this is an arbitrary threshold which we choose here, to a value of 0.85. So if each and every of these values here, so this means that phi k, j, l, and m, so all of them, all 4 of them, are larger than 0.85, then we would end up with crystals like this, which are pretty much ideal, I would say. So if you have only 3 of them in this large value and one of them is smaller, you basically squeeze the crystal somehow to get this type of flattened crystals here. So only 3 of these values are larger than 0.85. You can imagine how the story goes. We did this also how it looks like when you have 2. So you squeeze 2 sides, you end up with an elongated shape. And if you have only one of them larger than 0.85, which is basically the one which is per definition there, then you end up with pretty irregular shapes. So what we did now with this data set here, we took a process here, and this was a seeded batch cooling crystallization. I spare you the details of the experimental procedure. And we took samples every about 30 minutes and did a characterization of approximately 1,000 crystals per sampling time. So you end up with something like this, and the different lines you see here are the different shape types. So the ideal, the flattened, the elongated, and the irregular one. So you see here that the ideal shapes here increase, so this is kind of a healing process, while these irregular shapes here are depleted pretty fast. So why is this? When you could imagine that this is due to some phase-specific growth rates. We believe it is not. It is just some healing of irregular particles. You see here an irregularly shaped crystal as an initial value, and if you now just let this crystal grow with a phase-independent growth rate, so each one grows identically on the right side. So here you see the size normalized one. This one here is the non-normalized one, so this will actually grow. That started, so here, see this will grow, the other one will stay roughly the same, but you see that here, this is still the elongated shape, but this will then switch at some point with a flattened shape, and in the end, it will end up as an ideal shape. If you consider this as the major mechanism, you can start with doing a simulation, starting with this as the initial condition, and let it run by phase-independent growth rates, and you would end up with something like this, which is not too bad. I mean, we do not perfectly hit the values you see here, but the tendency pretty much looks like this should be the governing mechanism here. So let's get back to our stairway to heaven for some couple of minutes here. I think it's fair to say that you have a quite well-balanced modeling framework already to rigorously deal with crystal shape, either in terms of morphological population balance or with this type of geometric Monte Carlo methods. I think it's also, I tried to show you that with computer tomography, we can at least pick single crystals and agglomerates and in some unprecedented three-dimensional detail, tell them this is the shape of you. With increasing sample size, we're also able to track shape dynamics to some level of detail here, which I hope I can show you. And the huge thing which is still open is, from our wish list, is basically the connectivity here. So, and with that, sorry to say, I think we have to fall back to the Rolling Stones here. You can't always get what you want. I do not see any inline online capabilities of tomography on the horizon at the moment, but hey, maybe somewhere over the horizon, there will be a place where this truly becomes possible, but not for now, I would say. For now, I would say we have not yet reached crystal shape heaven, but we kind of have at least have been knocking on heaven's door. Nevertheless, I would say that also the techniques we have by now helps you a lot in studying further details of the system. You have seen that most of the results I showed you have been for potash alum. This is mainly because the geometry stayed relatively simple for the methodological development, and also it is easily handleable in the lab. But of course, you can go to other chemical systems. At the moment, for example, we're looking at paracetamol or lactose crystals. And if you have any interesting crystal shapes here in the right size, you can call me maybe. Phenomenologically, I would say there are also many other things that can be studied here, especially in the area of breakage and agglomeration here. So you can try to identify this broken face here. At the moment, we're not able to deal with these chipped off parts. This is something we want to develop in the future here. And here you have basically a combination of crystal, of attrition, of rounding of the edges here, and agglomeration here. Well, generally, I do not say that nothing else matters when it comes to crystallization, but I'm convinced that you ain't seen nothing yet when it comes to crystal shape. We will try to walk this way in the future, but for today, I have to admit that I still haven't found what I'm looking for. All this would not have been possible with significant help from many other people. First and foremost, I want to mention my PhD students, current ones, Simon Schiele and Felix Luxemburger. Also lots of the image analysis base algorithm have been developed by Tijana Kovacevic, and some of the theoretical foundations on convex geometry have been formulated by Alexander Reinhardt. Some of the work presented here today has been also developed in collaboration with the group here at TUM from Franz Pfeiffer, and also from the group at Kai Sundmacher from the MPI in Magdeburg. For the money, I would like to acknowledge the funding from DFG. We have seen this logo here already yesterday. Many of this work here has been funded within the framework of a priority program. Last not least, if you want to check out the Spotify playlist, I gave you about 19 different musical references with the hidden Easter eggs. And if you want to like to check them out, just follow this QR code. Thanks you for your attention, and I'm happy to take a couple of questions if the time permits. Thank you.
So, thank you very much, Heiko, for this excellent talk. We've got several questions. Can you hear me?
Yes, I hear you.
Yes, okay. And the first question is, what is the time for processing the tomographs? Connected with the question, why can't you use just confocal microscopy?
You can do confocal microscopy to some extent. You cannot get to concavity. Try to imagine doing confocal microscopy with this aggregate. You can probably get some information on the surface on this one here. I would say confocal microscopy is a three-dimensional surface measurement technology, but not around the particle. You can just achieve this by X-ray, I would say. Regarding the time, I was expecting this question. I was trying to drop the numbers during the talk. It's in the order of hours, I would say. It very much depends on the resolution you want to have. If you do a pre-scan to get a rough idea of the system, or you want to have really go to the limits of the system. The limits for our system here are, if we are very, very positive and everything works out fine, we reach an accuracy of about 1 micrometer. But this is already pretty challenging. And then we talk definitely about, let's say, 8, 9, 10 hours of measurement time of the system. Other things can be quite quicker.
Yeah, okay. And the next question was, what is the need to go in such detail that is complex and time consuming?
Well, it depends on if you want to advance science at this point. I mean, I tried to show you for the aggregation part, for example, you will not understand the aggregation process fundamentally if you do not go into this level of detail. I mean, this session here is called Fundamentals of Crystallization. And obviously, this is a fundamental interest. It is not something which is rightly applicable as a PAT system in a commercial process, certainly not. But you can learn much on your process when looking at this sort of detail, I would say. And I'd say that if you do not do this, you would end up with attributing physical behavior, which is actually governed by shape behavior. I have a couple of examples studied already where when you not consider shape very detailedly, you would ascribe physical behavior to a process, to an observation, which is actually shape-based.
And a further question from Joana Ferreira, TU 11. How can this study be extended to the early stages of nucleation? So that means when we have clusters, which will be later evolve in a crystal.
Well, we can go to synchrotron measurements where you can decrease the size to a certain extent. But when you really think of molecular clusters, I do not see that this is happening at this level of detail with the classical techniques which are around at the moment. We are a user of this type of micro CT systems. There is obviously development going on there. There is also at TUM, at the collaborating chair here, what they call a micro synchrotron, which can go to much higher resolutions, much higher brightness and detail than we can go. But still, if you really talk about molecular clusters forming, this is at the moment, I would say, is out of reach.
Thanks a lot. Maybe a very short last question from my side. Did I understand right that for most of these algorithms, you need the structure of the material you are working with? So you need to know this is potassium alum or like that, because you need the HKL values and so on.
This is a very good question. Actually, we realized that in the literature, for example, there is quite some confusion sometimes of correctly indexing with the Miller indices the different phases. And there are many errors actually which you can find in the literature. Mm-hmm. And this is, for example, what Felix Luxemburger does right now without any prior knowledge, but just based on attachment energy models to get an idea how this could happen and identify the phases without putting the information in there. So we're on this to do this without. At the moment, yes, we know what we are looking for, and we reconstruct phases, the smaller phases, on the basis of this knowledge. But for example, when you would consider a perfect potash alum crystal, you would never see something else than an octahedron. And we can already easily see this flattened, elongated type of crystals and so on. So observationally, this is already possible. In a very fundamental way, this is just what we're up to at the moment.
Yeah. Thanks again for your talk, Heiko. It was really excellent. And I think we need now to move to the next talk for today, which will be given by Yousra Tahri. She comes from University Claude Bernard Lyon 1 and LAGEPP. And when I understood right, this LAGEPP is a laboratory which is a joint laboratory of the University of Lyon and the CNRS. And also from the number of co-authors, we can see that this is obviously a collaboration project of different groups. And now it's your floor, Yousra Tahri, to tell us about your interesting work on L-glutamic acid and the rule of stages.
Yeah, thank you for your introduction. So my name, as you said, is Yousra Tahri. This work, do you see the screen? Is it okay?
Yes, we see.
Perfect. So my name is Yousra Tahri. This work was in a collaboration between many institutions in France, in Morocco, and Czech Republic. So today I will present to you our work on multiscale experimental study and modeling of L-glutamic acid crystallization. It's a new perspective on the Ostwald rule of stages. So the system that we studied is the L-glutamic acid, which is an amino acid that is used in pharmaceutical and food industry. Is a monotropic system that has two polymorphs, a stable phase beta, which has a needle-like shape, as you can see here in the pictures, and a metastable phase alpha, which has a prismatic shape. So we first studied the crystallization of L-glutamic acid polymorphs in a stirred crystallizer, 2-liter stirred crystallizer, which was equipped with a video probe. The video probe allows us to follow the crystallization through in situ images of the crystals, and also it was equipped with an invariant probe that allows us to detect nucleation. So we performed the crystallization of L-glutamic acid in water. It was a cooling crystallization, and we aimed to study the effect of supersaturation and temperature on the crystallization of the polymorphs of L-glutamic acid. So here in the graph, you can see the solubility curve of alpha, which is the metastable phase, and the solubility curve of beta. Each of these markers is one experiment. We always started undersaturated. We cooled our solution until a certain final temperature, and then we waited for the crystal to appear. And here you have some images of the crystal that we observed thanks to the video. So for example, for the experiment A, we only observed the polymorph beta. For experiment B, we observed mostly the polymorph beta with alpha. with experiment E, both polymorphs, and with experiment G, mainly the polymorph alpha. So actually these experiments permit us to draw a limit of crystallization domain for both polymorphs, and we could see clearly that at low temperatures, we had the preferential crystallization of the alpha phase, which is the metastable phase, while at higher temperatures, we had the preferential crystallization of the stable phase, which is beta. And we could say at the end that our system followed the Ostwald rule of stage only at low temperatures. Then we investigated the crystallization of L-glutamic acid in a thermostated 4 milliliter cell without agitation. This cell, we controlled its temperature using Peltier elements. So the cell and the Peltier elements were put under a microscope that was equipped with a camera, and we could take pictures of the crystals every 10 seconds to see what happened during the different crystallization experiments. So we investigated different operating conditions in the 4 milliliter cell. We started always undersaturated, then we cooled, and we waited to see which crystals appear. And for all the experiments that we did, we only observed the polymorph beta, but depending on the supersaturation, we observed different crystal habits. For higher supersaturation, the crystal habits was lozenge slabs that were actually at the beginning surprising because it was similar to the alpha phase, but actually no, the X-ray confirmed that these lozenge slabs were beta. And then at lower supersaturation, we could observe the usual crystal habit of the stable phase beta. So to sum up, normally we had in general large induction times for these experiments, but we never could observe the metastable polymorph alpha even at low temperatures. Then we continued our investigation of the L-glutamic acid crystallization in another scale in 500 nanoliter droplets in a microfluidic device. We generated the droplets in Teflon tubing by alternating the L-glutamic acid solution with oil. We stored our droplets in a thermostated sample holder, and this thermostated sample holder could be observed using a motorized camera. So actually we could follow the crystallization inside of the droplets during time. So for our experiments in the microfluidics, always we start undersaturated, and we go to the final temperature and we wait to see what we will have crystallizing inside of the droplets. So here it is important to note that in each experiment we had many tubings, and we stored more than 50 droplets. So each experiment has more than 50 droplets of so it's many nanocrystallizers. So in general, what we observed is that we mainly had the stable phase beta with some droplets where we could observe the metastable phase alpha. For example, here for this experiment, you can see a droplet with one crystal here, which is the alpha phase, and another droplet with the beta phase. So in some droplets, of course, we could observe both polymorphs, and even here you can see that there is some crystals of beta who were next to the crystals of alpha. So to sum up, in the stirred crystallizer, we have the crystallization of the beta phase at high temperature, and the metastable phase alpha was observed at low temperature. We deduced that the system obeyed to the Ostwald rule of stage at low temperatures, while in the milliliter cell, we only observed the stable phase beta, even at low temperatures. And this may be caused by the surface to volume ratio, which is high, that this promotes the heterogeneous nucleation, and the heterogeneous nucleation favor the apparition of the stable phase. Also, the volume is reduced and the mass transfer is weakened, so somehow the alpha phase nucleation was inhibited. In the microfluidic device, we mainly observe the stable phase beta with the metastable phase alpha in few droplets. And this could be because when we compare this result to the milliliter cell, is more or less the same, but there is only one difference, is that in the microfluidic device, the high number of nanocrystals increase the probability to observe the alpha phase nucleation. So the results in the milliliter cell and the microfluidic device, they are in accordance, but we don't understand why the beta phase didn't crystallize in a significant manner at low temperature in the stirred crystallizer. And we were like, why do we have these results with the stirred crystallizer? And for that, to understand the phenomenon, we went through modeling. We used the kinetic equation model. The kinetic equation model is based on attachment and detachment of monomers. We took into account in this model nucleation, growth, and the ripening. The ripening mechanism is induced by the solubility dependence on size, which is the Gibbs-Thomson effect. So we wrote the kinetic equation for each polymorph, and we took into consideration the differences between the two polymorphs, which are mainly reflected in the shape, the solubility, the interfacial energy, and the growth rate. So the first interesting results that we got is concerning the size distribution of the clusters. So we did the simulations at 5 degrees, which is a temperature at which the system obeys to the Ostwald rule of stages. And here you have the size distribution of the metastable phase clusters and the size distribution of the stable phase clusters. What is striking here and important to see in these graphs concern the stable phase, because if we see at the time of 0.05 seconds, the critical size was around 3 nanometers, and you can see from the number density that we had a certain number of crystals, of clusters that were supercritical. It means they had a size that is higher than the critical size. Then at 1 second, the critical size is almost 10 nanometers because the supersaturation was consumed. And here we can see that all the clusters that we have are under critical. So to better understand this behavior, we tracked the crystals that are larger than the critical size with time. And for that, we introduced the crystallization fraction for both polymorphs. And here we can see clearly the evolution of these clusters. So here we can see that this fraction goes until a certain maximum for alpha, then goes back to a non-zero value, while for beta, it goes to a non-zero value and then fade to 0. So here we can see clearly that it was a disappearance of supercritical clusters or crystals of beta. So how can we explain this? So normally both polymorph crystallize more or less at the same time. Then when the alpha phase has a fast growing phase, will grow very fast, will consume supersaturation. Also the stable phase will grow, but much slowly. The supersaturation is consumed, the critical size of both polymorph will increase, and at some point the stable phase will find itself undersaturated and will dissolve. So this permits us to have a new perspective on the Ostwald rule of stage in case of fast growing metastable phase, is that in the nanometric scale, both polymorphs crystallize at the same time, but because the metastable phase grows much faster than the stable one at some point will find itself undersaturated and will dissolve, and what we will observe at the macroscopic scale is only the metastable phase. So to conclude, in the stirred crystallizer, we observe the crystallization of the metastable phase alpha at low temperature. In accordance with the Ostwald rule of stage, And we could explain this by using the Gibbs-Thomson effect, which is a pseudo Ostwald ripening between the two polymorphs. In the milliliter cell, only beta crystals were observed. This is because of the promoting of the heterogeneous nucleation. Also, the Gibbs-Thomson effect between the polymorphs is low because there is no agitation, so we don't have this competition that is promoted between the two polymorphs. And in the microfluidic device, also we observed mainly the beta crystals because the Gibbs-Thomson effect is not promoted, but the high number of droplets increase the probability to observe the alpha phase. So I want to thank the region Rhône-Alpes, the Crystallized Cost Action, and the Ministry of Education of Czech Republic for funding. Thank you for your attention, and please feel free to consult our articles for more details. Thank you.
So thank you for this very interesting talk and this very systematic experimental work you have done. And my question is related more to now practical application. Do you see any conclusion for crystallizing polymorphs now from these new aspects you introduced us?
Yeah, I think that we usually, when we model polymorphism, we neglect this effect, this Gibbs-Thomson effect, this ripening that comes from the Gibbs-Thomson effect, and I think that we should find a way to take into consideration this ripening in the modeling, that the outcome can really change. My simulations, our simulations, they concern only like very small clusters. We didn't go to the macroscopic scale, but it would be interesting to have a model that goes until the macroscopic scale, which take into consideration the ripening mechanism between the polymorphs.
Mm-hmm. In fact, which was the objective of this study when you started?
It was to understand why, in some conditions, we couldn't have any clusters of the stable phase, and we had only the metastable phase. Especially, like, we had this look at we were thinking about it, and we said maybe at some point the metastable phase, when it's fast growing, in the case of fast growing metastable phase, at some point we can have the small clusters of the stable phase that will become undersaturated and dissolve. But how can we show this? Because experimentally it's difficult. So we try to do experimentation on different scales, and then to model and to see what happened.
No, thanks. We have some questions from the first one from Antonia Borisova. She's asking, how did you determine the critical size of the clusters?
How there is an equation for the critical size. Is from the work energy that is like is the work of like is the energy. Normally there is the critical. I'm sorry, I am getting confused. So normally is from the activation energy. That's it.
Okay, let's go to the next question, which is why not using simple population balance equations with size-dependent solubility to describe the ripening process?
Yeah, actually, this is a very relevant question. We tried to use the population balance model, but the population balance model is not adapted for such a system because I will show you a slide. We have, we have seen that actually we made a population balance model that normally the clusters will grow if they have a size that is higher than a critical size, and they will dissolve with a size that is lower than the critical size. and we came to the point that normally the nuclei are born exactly at the equilibrium crystal size, and actually they cannot grow. So this model is not cannot take into consideration the ripening mechanism that is from the size dependent solubility.
Okay, and I see there is a further question, which is it possible that a different polymorph would be obtained if the crystals are dissolved and recrystallized?
Can you repeat the question, please?
Is it possible that a different polymorph would be obtained if the crystals are dissolved and recrystallized?
The question is about which the experimentation or the modeling, and in which device?
I think I understand it in a way that it's asked if you have a dissolved, you have a solution, an unsaturated cool down to get nucleation, to recrystallize, and then is it possible to reach or to get a different polymorph?
It's so you said when we can get a different polymorph depending on the operating conditions, or because we repeat the same experiment. Yeah. If we repeat the same experiment, normally we get the same outcome, but if we change the operating condition, then we may have something else.
Yeah, I think also for this system, it's known that there are only two polymorphs.
There are only two polymorphs, exactly.
Maybe another one can appear in completely different conditions. So thank you very much. So I think we have to move to the next talk. Thank you for this systematic work.
Thank you.
Yeah, now let's move to the next talk. The next talk will be given by Hendrik Rehage. He is a PhD student in the group of Matthias Kind at the KIT, the Karlsruhe Institute of Technology in Germany. And he will speak today about a new method for scale-up of mixing-controlled precipitation processes. So, Hendrik, we are eager to listen to your talk. The floor is yours.
So, hello, everyone. Can you see me, and see the laser pointer?
Yes, everything is okay.
So, thank you very much for the kind introduction. My name is Hendrik Rehage, and I'm here to present you a new method for scale-up of mixing-controlled precipitation processes today. On my side, thank you very much for your attention. We did not publish any of these shown data yet, but it may help you, like we noticed some similar effect for competitive parallel reactions. So you can maybe have a look on these publications here. And if you have any questions left, feel free to ask in the Q&A session right now. Thank you very much.
The last talk will be given by Gunjan Das. She is a doctoral student from the University of Leeds in the UK, and she will speak about new in situ. Yeah, there she is, in a new in situ X-ray phase contrast imaging microscopy technique for continuous antisolvent crystallization application. So it's your floor, Gunjan.
Thank you very much for your kind introduction. Good afternoon, everyone. My name is Gunjan, and I'm a PhD student at the University of Leeds, working with Professor Sven Schroeder and Professor Jan Sevcik from CMAC, and collaborating with Diamond Light Source. Today, I would like to talk about real-time phase contrast imaging of continuous and antisolvent crystallization using a concentric flow microreactor. Before I start my talk, I would like to give a quick overview of the things I'll be discussing today. So I will start off with introducing the context of the study that we performed. Then I would like to introduce the technique we have used, which is known as X-ray phase contrast imaging and will be referred to as XBCI. We have looked into glycine and lovastatin crystallization, so I will be talking about what we have observed and the key findings. So, continuous antisolvent crystallization. Why would we study this? This can provide uniform flow and mixing, which can lead to consistent and repeatable results, the equipment cost and the device cost is very inexpensive, which really attracts the chemical and the pharmaceuticals industries, as well as it can actually solve the problems with batch-to-batch variations, as well as scaling up. However, there are some challenges associated with this, which is nucleation is actually dependent on complex local mass transport and phase dynamics. It potentially has complex fluid dynamics as well. There are also unknown local supersaturation and liquid-liquid phase boundaries, and there is an influence of mixture geometry and wall materials. So this entire process is actually spatiotemporally complex, and one needs a microscopic imaging technique with phase and chemical sensitivity. Many techniques have been actually applied, but not all of them have been able to provide the required information, and there have been also limitations with experimental setup. The ideal technique should provide structural information and also imaging insight, provide information at a molecular level, and have some spatial and time resolution. As we can see, not all the techniques shown on the table tick all the boxes. However, synchrotron-based X-ray imaging ticks all the boxes, making it an ideal technique, especially considering nucleation is a multiscale process, and it's a rare stochastic process, so this would be a very suitable technique. As many of you would know and familiar with laboratory-based X-ray imaging, however, when you're studying organic phases, the data acquisition would be very slow. It could take between 6 to 8 hours, and also the contrast is extremely weak. However, with synchrotron-based X-ray imaging, there is some flexibility and there is some advantages. For example, you can get high contrast. You can get very beam size, so the beam size can be actually varied depending on the material you'll be studying, and that could be changed and altered. You can have high energy beams, meaning you can the X-ray beam can penetrate deeper. You could have higher photon flux brilliance, meaning the data acquisition will be much shorter. You can get higher resolution, and of course, there are multiple contrast modalities available, and which one we have used is X-ray phase contrast imaging. The principle of XBCI works. So as you can see, you need coherent X-rays, and that is only available at the synchrotron facilities. Now, for this technique to work, how it works actually is every material has a variable refractive index. Upon interaction between the sample and the beam, the X-ray beam, there is a phase shift that's being introduced, causing an enhanced contrast, which is actually also caused by constructive or destructive wave interferences at the interfaces. The technique we have used is propagation-based XBCI, where the cameras move to different distances comparing to the sample for an optimized contrast. So how does this work in practice? The experimental work has been carried out at I13-2 beamline at Diamond Light Source. We have performed fast imaging paired up with XBCI. We have taken roughly 20 frames per second, and the aim was to visualize crystallization with a chemical contrast. So what we have done is we have performed imaging inside the crystallizer here. So speaking of the crystallizer we have been using a concentric flow antisolvent microreactor, which is initially designed by the Sevchik group. And what we have done is we have modified it with an outer Kapton tube for X-ray transparency. What we had the solution with the API, or glycine or the lovastatin in the case that we have studied, flowing through the inner tubing. And what we had flowing through the outer tubing was antisolvent, which would force the crystallization to take place. What we have also done was we have taken X-ray imaging at the end of the inner concentric tubing and the beginning of the mixing region. The solution was the resulting slurry solution was collected later on to perform to collect the crystals and perform some tomography. So glycine imaging study. What we were able to see. So this is the inner tubing, as I showed in the previous image. And what we had, the glycine solution flowing through the inner tubing and the antisolvent flowing through the outer. We were able to observe the liquid-liquid boundary, and we were also able to see 3 of these lines. And that the reason behind this, because the inner tubing was flowing at a much faster rate comparing to the outer tubing, so which causing the gravity was forcing the outer tubing liquid to move slightly more slower and backwards. And also we were taking 2D projection of a 3D process, leading to these lines being there. So these liquid-liquid boundaries we have seen is because we, like I said, we're looking at a 3D process. So the boundary closer to the camera appears to be much darker than the boundary further away from the camera appeared to be much lighter. So as you can see, there's an orange box highlighted. That's actually the region we're mostly interested to look at, to understand and find out more about the molecular structure and the phases in that mixing zone in particular. And I'll be showing you the video we have obtained in a minute. So here's the video, as you can see. And what we can see is that we can see the inner tubing flow and these boundaries as well. You can see here the heterogeneous nucleation of a crystal, which just got flushed away due to the high flow rates we have. As you can see, these boundaries actually are always there, and you can see them moving back and forth, of course, simply because the imaging was done very fast. What we will see in a minute will be homogeneous nucleation that will be taking place in this corner of the video. If you look at it now, you also see a lot of mixing, and you can see some crystallized crystals forming towards the end, this right-hand side. So yes, so as I've said, so here, this is what the heterogeneous nucleation looked like. What it's done was, it due to the high flow rate, it got flushed down, causing secondary nucleation downstream when we've done some imaging down there as well. What we have also done was we have collected the resulting slurry of glycine solution, and we have performed a quick tomography to see the microstructure and to see the crystal habit of glycine crystals. We have also done antisolvent crystallization studies of lovastatin. As we know, lovastatin always crystallizes as needles, and we were able to observe a lot of heterogeneous processes taking place on the outer walls of the tubing. When we were watching crystallization real time in situ, we have seen the needles here where I'm pointing out, we have seen the needles growing. And what we have done is we have actually also performed a tomography as well, where we have done a 3D image, and we have seen them as well. Now, needles in industry or even in any labs are a huge problem because it causes difficulties with filtering, poor flowability properties, and of course also with pressing them into tablets. Now, what we were able to observe might be actually helpful in the future. We were able to see those heterogeneous processes that I have mentioned a minute ago. And what we can see them on the very right-hand side, we see the gray shape, which have sharp edges, suggesting it has a crystalline-like character. However, we do not see needles there. I will show you a video that we have, the real-time imaging we have done. And in the video, we have done a number of processing here to enhance the visualization. Right, so what you can see now is you can see these 2D phases growing here, and what you're also able to see some spherical droplets join and coalescing together, causing oiling out effect, which I will speak about in a minute. But the main fact is these 2D needle-like 2D phases do not have a starting point like needles. So maybe something can be done in the future to modify and avoid this needle-like growth behavior. So this is the image I was talking. This is one of the still image taken from the video. We can see some spherical droplets joining together, and like these spherical droplets here, they join together, and eventually they form this thin film edge, which then later on, when we watch the video, we can see the needles growing. One thing I also didn't mention in the video earlier, because things were much happening in a very faster speed, there were also at the same time there was some homogeneous nucleation taking place, and they were appearing as plain white little pixels or dots in the video. So my summary is we were able to observe potential 3 pathways to nucleation, and they were, one of them was via an oiling out path where these spherical droplets were joining together and then they're coalescing together, leading to heterogeneous nucleation. The other pathway was through the 2D film phase, which is caused by, since lovastatin is such a huge molecule, it started, they started forming due to the attraction between the outer reactor tube and the actual molecule. And the final pathway was the homogeneous nucleation, which we have observed as white dots in the still image I have shown you. And all these 3, of course, lead to the crystal growth. Another thing I would like to mention is X-ray imaging is an extremely powerful tool, just like a Swiss Army tool, because while we're taking imaging at the same time, we're able to obtain a number of information while we're taking one single scan or doing one single crystallization study. What we're able to see was, while we're watching, we could see nucleation, crystal growth, we could observe the microstructure, phase separation, film deposition, solvent mixing, and kinetics. So the ongoing work we have planned is, and we're doing right now, is we would like to continue furthermore with lovastatin studies using the XPCI technique, and we'd like to test and try it with different concentrations and different flow rates to see how these 2D phases grow and to observe how these heterogeneous nucleations actually behave under different conditions. But we also would like to use synchrotron-based different imaging modalities, which is diffraction imaging and pair distribution function imaging, to actually focus on the areas highlighted in orange on this slide. On this note, I would like to thank all of my collaborators, and thanks to all of you for listening and your time and attention. Thank you.
Thank you very much, Kunyandas, for this inspiring talk. I think we could really see that this synchrotron-based technique is a really powerful tool for online monitoring crystallization processes. My question, since there is no other at the moment, is I'm wondering that you observed oiling out. Because oiling out always means there is a miscibility gap in the liquid state. Is the phase diagram known for that system? So lovastatin in the solvent you used?
I personally haven't looked in this yet, because we're still trying to explore more and understand. This is something we have obtained, and that's something we definitely would need to look into. But I can refer to my supervisor later on and come back to you if that's okay.
Maybe, yes, of course, that would be interesting.
Yeah.
Because oiling out is mostly a phenomenon which is not, which we do not have, because we can have crystallization of the wrong phase, for example. But now there's a question I see. Wasn't the scale too large, 300 micron, to see primary nucleation? How big are the nuclei in the picture?
So if we go back to that slide, so that was actually 100 microns, that the scale bar that we have. So I would say maybe just, it wasn't that huge.
Mm-hmm. So I see there is no other question. Then I would like, oh yes, there's another one. Question coming. Is the resolution that you have in your images above 1 or 2 microns range?
Oh, so what we were trying to achieve, so using all the detectors and the devices we have used and the optics, we should be achieving about 0.81 microns. But due to the experimental limitations, we have definitely achieved 1 micron, but we are working towards getting under that.
Nice, and I see a next question from Bart Zweinenburg. He says, a very nice presentation, and you will look at the structure of the heterogeneous crystals and nuclei. Can you already rule out contamination as source of these particles?
Sorry, I didn't get that.
Was there any impurity in the solution? What could be a contamination leading to these crystals?
No, it shouldn't be, because we have used everything was sort of in situ. It was a continuous process. However, what could have caused this? We used the outer Kapton tubing, which was welded, so that had some that had some points where it could have caused sort of nucleation points and crystals to grow.
Now I see a comment from one of your colleagues, Sven Schröder. He writes, we are not seeing primary nuclei, we see sub-micron crystals. This is, I think, clear due to the resolution. Okay, then I would like to thank you. And with this last question we had, I would like to close the lecture, the session for today. I would like to thank all the speakers and also the participants asking questions, and let the floor for the organizer for the closing remarks by Marco Mazzotti.
Thank you very much, Heike. And I would like to share my screen
I will stop here.
Yes. So I would like, first of all, to thank all the speakers. So I would like to thank Heiko, Yousra, Hendrik, Gunjan, and of course you, Heike, for chairing the session. And I would like to thank all participants for the active participation and the questions. Tomorrow, there is the third day of this webinar series. The focus will be on industrial applications. You see the list of speakers here. The webinar will be at the same time as today, and I look forward to seeing many of you tomorrow again. Thank you very much, and good evening and good day, depending where you are on the planet. Bye-bye.